Dr. Doudna, thanks so much for joining us. We're really excited to have you here today. And the theme of our conference is breakthrough innovation. And of course, your breakthrough innovation is a big one. So we're very excited to hear from you. And I thought maybe just to start off, you could tell the group, kind of set the stage for us. You know, what is CRISPR and why is it so important?
Well, it's great to be here virtually. Thank you so much for inviting me. CRISPR is a technology for editing the code of life, editing DNA in cells. And as you heard briefly in the introduction, it's a precision tool. So it allows scientists to do that kind of editing with a level of accuracy that until recently, was really not possible. So it's really transformed not only scientific research in biology, but also the kinds of applications that we have at our hands now that we have this technology available.
That's great. So maybe you could help us think a little bit about the ingredients that lead to a breakthrough like CRISPR. This is a crowd that's very interested in having breakthroughs in their areas and in their fields. What do you think the ingredients were? Like, how did this come about?
CRISPR is a great poster child for curiosity-driven research. It's a fascinating story because it really started, maybe 15 years ago at least, when a few scientists around the world noticed that bacteria have an interesting mechanism for protecting themselves from viral infection, a mechanism called CRISPR that involves a strategy for capturing little pieces of viral genetic material and using it to effectively immunize cells against future infection. And in my own work, I've always been interested in fundamental biology and in particular in how systems evolve to use molecules called RNA that can play a wide range of roles in modern cells, but are also thought to have really been in many ways responsible for the origin of life on our planet. So we began researching CRISPR and got into a wonderful collaboration with Professor Emmanuel Charpentier. And that work led to our understanding of how CRISPR-Cas9 works as a molecular scissors that is truly programmable. It can be programmed to interact with different DNA sequences. And that's really what makes it a powerful technology. So it was a project that started in one place and ended up in a very different place.
And when you think about some of the building blocks that, so you mentioned some of the collaboration, you mentioned, prior research, Newton said that if I've seen farther than others, it's because I've stood on the shoulders of giants. I'm curious, like in this case, do you think that's true of the CRISPR research, and are some of those themes true there as well?
Definitely, absolutely. I think CRISPR technology came along at a kind of an opportune moment when many in biology were aware that we have at our fingertips genome sequences that span everything from bacteria to yeast to mice to humans, and we have increasing information about the genes that are encoded there and what their functions are. What was missing was a tool to manipulate those genes, how to change them precisely, and that's where CRISPR comes in. So I've often thought that if CRISPR came along earlier than it did, it might not have had the same impact, because we really needed to be at a point where we had that level of genetic understanding and access to sequencing data to make use of a tool like CRISPR.
Can you talk a little bit about your research lab? And I think you've run a lab at Berkeley since, I think, 2002. And that was you led that group, and that was the group that made this discovery. Can you talk a little bit about it and sort of set the stage for that? And would love to then talk a bit more about what you look for in hiring and culture and things.
Well, I'm sitting here in my office in Berkeley, and I also founded the Innovative Genomics Institute a few years ago. So we're actually in our institute right now. And this is where my research lab operates. So I run a lab of about 40 people, and they range from students who are just starting college to very experienced scientists who have already received their Ph.D. and are in the lab here to get some additional training. So it's quite a range. And it's a really interesting, I think, place to do science here in Berkeley, because we have, we're at one of the world's best research universities. So we attract some of the very best students from all over the world who come here to work. And at the same time, we're literally across the bay, and I'm looking there right now towards Silicon Valley, where we have a lot of tech companies and entrepreneurs who are doing extraordinary things in hard tech. And so I think that junction has really, in many ways, spurred the entrepreneurial spirit here, as well as to allow us an opportunity to collaborate as appropriate as the technology of CRISPR has expanded. So, for example, we're now doing research that includes not only studies in animal models of human disease, but all the way to using CRISPR to understand, essentially create a dictionary of information about the human genome that will be accessible to machine learning in the future.
When you're hiring people to work in your lab, what do you look for? What characteristics matter to you?
Well, I always think about the very first person that I hired to work in my lab almost 30 years ago. She's still with me. Kai-Hong Zhao has been my right-hand woman in the lab for a long time. And when I first interviewed her, I was struck by two things. First of all, she didn't know very much about the science we were doing. It wasn't her background. However, she was incredibly passionate and incredibly energetic. And she said to me, "Jennifer, I'm going to do whatever it takes to make sure that our science is the best." And that's still true almost 30 years later. And so I think, she's somebody who exemplifies the kind of person that I look for. They don't have to necessarily come in already with the expertise they need. They can, I feel, smart, motivated people can learn that. But what you really need is you need people who are great team players, who really want to work together, who want to swing for the fences, try doing things that are hard, that other people think won't work, because usually that's where the real breakthroughs happen.
And when you think about the culture of the lab you're trying to create, what are the kind of key tenets there, and how do you go about creating those?
Well, it's very interesting, because when I think about this a lot, because when I started my laboratory in 1994, of course, it was a much smaller group of people at the time, but science was different in those days, I feel, too. It was very much more the case that individual members of the laboratory were working on projects that did not necessarily intersect directly with what other people in our research group were doing. So they were, very much kind of free agents that are independent workers in a way. And then what's happened over time, especially I would say over the last decade, is that biology has really changed. Now I think the most interesting kinds of questions that we can try to answer are addressable only with a complex combination of approaches. They include not only being able to do molecular biology and genetics, but also knowing something about computer programming, being able to analyze large sets of data. And so as a result, I've really encouraged a spirit of collegiality and collaboration, even just within the lab. So when new people come to the laboratory, I encourage them to talk with their colleagues, hear about what other people's projects are, figure out where they would like to start working and contributing. And what I've often found with that approach is that people, collaborations even within the lab will happen organically. People are drawn together, they start working in little groups on problems. And it benefits everyone. It benefits the science, and I think it makes our work more fun.
I mean, to your point about all the different areas that have to kind of come together to make successful research, how much do you try to map things out versus let things happen and bubble up? There's clearly a tension there. It's a tension that I think a lot of data science leaders struggle with. How do you think about that in your organization?
Right. Well, not surprisingly, maybe I try to do both. So, there are certain problems or ideas that we have that are, very exciting that we want to pursue. And for those, I'll try to hire people that I know are interested in working in that area and to help them get connected to the folks that they're going to be able to collaborate with on projects like that. And at the same time, I always encourage the folks that are coming into the group to think creatively about ideas that they want to pursue, because I don't have all the great ideas, and I don't think any one person does. So we really need everyone to be thinking creatively about what we can be doing, given our expertise, given the cutting edge of the field. Where can we be working most productively? And what I've found is that in many cases, those really creative, really interesting ideas come from unexpected directions. They come from, say, a summer undergraduate student who comes to the lab who isn't biased in any way, and they have an idea that you say, wow, gosh, I hadn't thought about it that way before, but that's very interesting. And it's just, I think, and then I always kind of look for the opportunities to bring those two approaches together. So in other words, if somebody comes up with a really interesting idea and they want to work on it, I think it's worthwhile, I will, do whatever I need to get them the equipment they need, the resources, and importantly, the right collaboration, if that makes sense.
Absolutely. And how do you promote sharing within the lab? Is it more, do you find informal mechanisms are more important, people bumping into each other in the hallway, or do you try to organize, sessions and conferences and things like that? Or I'm guessing it's some combination of both, but I'm just kind of curious how you think about that challenge.
Right. Well, we have, the usual Slack channels and things like that to allow people to share in real time their ideas and their information and their data. We also have a series of different levels of meetings that we have. I don't like meetings in general, so I try not to have that too big of our sort of lab scheduling mechanism, but we do have a large weekly meeting of the group where we have a rotating schedule, people presenting their data, their ideas. And then I think maybe even more importantly, we have a series of what we call subgroup meetings, where we have subsets of people who are grouped together by their more detailed research areas. And those are really fun to attend because they're not very formal, and they're often just somebody opening their lab notebook and saying, here's what I'm working on this week, and I'm struggling with this problem, or, I'm trying to figure something out, or I just got some new data, help me think it through. And that's where we really have a lot of really smart people kind of noodling on data together. That's really a lot of fun.
And in that setting, like how big is that group usually? And I'm taking it you attend as well and problem solve with them?
I attend many, but not all, and I think that's also important. I've sort of set it up so that those are ongoing groups that get together and they carry on whether I'm there or not. I'm probably there 75, 80% of the time, but even when I'm not there, these groups are cohesive and working together. And they're set up pretty informally. We had to make them virtual during the last 2 years, and amazingly that kind of worked. People would carry on. They would be typing ideas into the chat during Zoom sessions and things. But of course now we're back to in person, which I think everybody prefers.
Yeah. So we're talking a lot about collaboration. In Isaacson's book about you, there's also a lot of competition in science. And I'm curious how you see that tension, both within your lab and also just in science in general. Like, how does competition play into all this?
Well, it's a key element. You're right. It's there, and I think in the book, there's really this interesting discussion about the tension between collaboration and competition. And I've always found that, I guess the way I think about competition is I always figure I'm competing with myself. I'm competing with myself to do the very best work that I can do, or that I and my colleagues can do together. And that's what I tell my group. So We always focus on high quality, what are the impactful questions that we can be trying to address right now. And other people are working as well, and there's lots of smart people out there. So we just try to stay aware of what's going on, but not let it influence us unduly, if you know what I'm saying. I'm basically, I'm trying to encourage members of my lab and myself to not be too influenced by other people, because I think innovation requires that you are trying things that maybe the crowd isn't thinking about or isn't doing. I mean, certainly CRISPR was that way in the beginning. But at the same time, of course, there's, sometimes there are ideas or discoveries that are made by other groups that absolutely influence your thinking, and they should.
Mm-hmm. Coming back to the kind of the theme of breakthroughs a little bit, one of the sort of popular images of breakthroughs is they come in sort of these like flashes, like Newton getting hit on the head with an apple or something. I mean, as a very accomplished scientist, how accurate do you think that is, or do you think science is more iterative, and how do you think about that in terms of structuring your research process?
I think there's something to that, in the sense that I think a lot of times in our field, in our line of work, that breakthroughs will come, if not exactly in a flash, in a very short window of time, because you sort of have a collection of data and ideas that converge, that make you suddenly realize something that you hadn't seen or thought about before. And I've had a handful of those experiences in my career. We all live for those kind of moments of discovery, I call them, where things kind of all come together, and it's an incredible feeling of joy when this does happen. And and it's just an interesting thing, because I think you have to, again, in our field, and I think this is true in a lot of areas of technology as well, that you have to have a certain amount of data, of understanding, to get to a point where you suddenly say, oh, this plus this thing over here means that we can now either do something incredible that's exciting, or we understand something new that we didn't understand before. And it's hard to predict exactly when those moments are going to happen, but it in my experience, it always involves kind of this convergence that suddenly kind of makes you realize something new.
Do you find yourself impatient for those moments, or do you have to teach yourself to be patient for them? Like how frustrated can you get, and is that a part of how does that fit into the process?
Yeah, well, that's also a great question. I'm always impatient. I'm very impatient. I always want those moments to arrive sooner than they do. But I think that's part of the process. It's part of the hunger. You have to be a little bit desperate in science. And this is probably true again in other in engineering as well, that you really need to have a deep desire to understand something or to do something, because a lot of these the kinds of really cutting edge things or breakthrough kinds of ideas that happen are they're hard, and they require risk. They require trying things that a lot of people say, well, it's not going to work, or that's not interesting. And I think back on my career, and if I had listened to those voices all the times that people said to me, oh, that'll never work, or that's not very interesting, I certainly wouldn't be here right now. So you really have to kind of trust your gut at some level. If you think something is worth going after, you just single-mindedly focus on that.
Coming back to CRISPR for a minute, what where would you say we're at in the CRISPR journey? Or are we in the second inning or the ninth inning, or somewhere in between? And how do you see this playing out from here?
Well, closer to the second than the ninth, that's for sure. I think that's an important point to make, actually, that the CRISPR revolution is really just beginning. I think we're at a point where the technology is, widely used around the world today in research laboratories, It's already producing exciting clinical results in certain types of disease. There's some really interesting applications in agriculture that are starting to be announced. But it's really early days. The technology itself is still under very rapid development, and then all of the opportunities to use these precision editing tools in DNA are still coming to the fore. And I think in the future we'll see wide-ranging applications that span not only clinical medicine and diagnostics, but also using CRISPR in microbial populations to address challenges of climate change, for example. I mean, I think they're just really important for people to appreciate that this technology really spans all of biology. Anything that has DNA is subject to CRISPR.
What are the big obstacles right now in terms of that more scaled impact that you're talking about for CRISPR?
Well, I think it's a combination of things. I think there are some companion breakthroughs that need to happen, and the big one that's on my mind right now is how we deliver CRISPR molecules into cells precisely. How do we do that in a patient? How do we do that in a plant? How do we do that in a soil microbe? It's all at some level it's all the same problem. It's how do you solve it? And it's a hard problem, and it doesn't just pertain to CRISPR. This is kind of the classic delivery problem, we call it in biology, where you have to figure out how to find the cells that you need to get your molecule into. And that's true whether we're talking about CRISPR or whether we're talking about a small molecule therapeutic. It's kind of the same challenge. So there's a lot of work going on there, but I feel that innovation is still needed. We probably don't, it's probably not going to be a one size fits all kind of approach. We're going to need a whole, panel of different delivery strategies that will allow scientists to effectively and precisely edit cells in whole organisms with, with an ease that today, isn't possible. So that's one example. I also think that there needs to be a really good sort of close partnership between independent and nonprofit organizations, whether it's universities or institutes that are doing research, kind of fundamental research in the field, coupled with companies and investors that can fund teams to develop ideas once they reach a point where they're clearly poised to have impact, but it's no longer at a scale where you can reasonably do it in an academic setting. You really need to have the resources and the focus that companies can put to a problem. And so that's one of our goals at our institute, actually, is to really try to make those partnerships as seamless as possible, and to also encourage young entrepreneurs who are coming through an organization like ours, and in many cases get very excited. They may make a fundamental discovery themselves, or they may have an insight while they're working here that makes them realize that they have a way to solve a problem. And then we try to give them the resources, whether it's financial or other things, to help them be successful with those ideas in a for-profit setting.
When you look forward 5 or 10 or 15 years, you can tell us the right time frame to think about. What kind of impact can we expect from CRISPR? I mean, what should we be looking forward to, and on what time frame?
Well, I'll preface that by saying I'm really bad at predicting the future. I have no idea what's going to happen even next week, really. But that being said, I think with CRISPR, certainly within the next few years, let's say 5, I really think we're going to see increasing, approvals for CRISPR-based therapies, probably initially for relatively rare diseases that are in the bucket of things that are being tested right now. Sickle cell disease comes to mind, and there are a number of rare genetic diseases that, CRISPR is actually a great technology for that, because it can be tailored to, even to individuals if necessary. So I think we're going to see, increasing pace of those kinds of therapies rolling out. But I think in the long, and I should say also, kind of going hand in hand with that is, diagnostic approaches that take advantage of some of the CRISPR chemistries that are more recent, that are very interesting for detection of disease-causing organisms, but also detection of disease-causing mutations in tissue. I think that's a really interesting area to keep an eye on as well. So those two things over the next few years. But maybe if we're talking longer frame timeline, like over 10 to 15 years, I'm actually very interested in the possibility that CRISPR becomes also a preventive type of, I don't know if you call it therapy, but for preventive healthcare, where you could imagine having access to DNA sequencing data that I think in the not too distant future we'll all have it for our own DNA, and instead of that being nice to have and gee, it's too bad I've got this susceptibility in the future, CRISPR can then come in and be utilized as a way to prevent disease by, tweaking genes that instead of making us susceptible to disease can actually be protective. And this is already happening. I mean, there's at least one company out there that's working on this type of a strategy, and certainly lots of nonprofits and academic labs are working to do this kind of thing, because I think that's where CRISPR ultimately will have very broad impact, is if it can be used in preventive medicine.
You mentioned earlier the institute you're building. Can you tell us a little bit more about it and how you see it sort of sitting in this whole ecosystem of nonprofits and research and startups to catalyze what you're trying to achieve? I mean, one of the things that we're very interested in the conference is scaling things to impact, and it seems like that's kind of the goal of the institute. So I'm kind of interested in how you're thinking about that.
Right. I think that is such an important topic. It's very interesting, and it is on my mind a lot these days, because CRISPR is a great example of something that started very, very small, really mom and pop kind of science. And now, it's huge, and it's going to get even bigger. It's going to get much, much bigger. And it needs to have the kind of global impact that I think many of us anticipate, but we're not there today. And so our institute, the Innovative Genomics Institute, was founded with the idea of science with social purpose. And what do I mean by that? Well, again, for me as a scientist, I'm very excited about CRISPR. It's it's interesting biology. It's a wonderful technology. I'm also aware that it needs to go from where it is today to, it really is an issue of scaling it to a point where ultimately you'd like anyone to be able to benefit from CRISPR who needs to, whether it's because they have a genetic disease or because they're trying to grow crops in a drought susceptible area, or anything else where CRISPR can have an impact. How do we do that is the question. And so at the Institute here, we're really focused on problems where we think we can have real world impact by looking not only at ways to develop the CRISPR technology and its application, but also doing that with an eye towards scaling it and appropriately partnering it with companies when it gets to that point. And I'll give you one specific example, and that is that, so we're very proud that we have an FDA approved investigational new drug, or IND, study ongoing for sickle cell disease. I think we're the only nonprofit organization that has this. And so we're not, companies are doing this, but we're doing it as a nonprofit. And why are we doing this? Well, it's a partnership between 3 of the University of California campuses, Berkeley, San Francisco, and Los Angeles. And so we have 2 of the world's best medical schools involved, and partnering with Berkeley and the Innovative Genomics Institute that has, kind of is a real engine for the CRISPR technology. And so our idea here is to get this trial going, but we're doing it with an eye towards ultimately figuring out a way to deliver the CRISPR molecules into these patients without requiring a bone marrow transplant, which is what they need to go through today. That will have a huge impact on cost and obviously have a very large impact on the ability to scale this approach in the future. And so we can do this as a nonprofit. We have money from the state of California. We have money from donors. We have money from the government to work on this. And we have a lot of very enterprising, young, smart students who are very motivated by this kind of a project. And so our goal is to get to a point with this work and this trial that we can then find the right partnerships that allow us to, for example, have a tiered pricing structure when that becomes appropriate, and to help us get over the finish line in terms of getting this all the way to FDA approval. So it's a process, and we're doing this with a number of our other projects as well. It's just a very exciting way to have kind of this ecosystem of innovation that's coupled to teams that know how to scale when the time comes.
How do you think about the role of startups in the ecosystem? I know you're involved in some, or the Institute's involved with some. How do they fit in?
Yeah, they play a big role. Startups are in many ways, have become a very important outlet for many of our trainees. They're, students that come through the labs here who realize that they have a really exciting idea or discovery that they've made that is appropriate to build out in the context of a company. And we really encourage that. We've got a couple of programs running here at the Innovative Genomics Institute that support entrepreneurs. Tory Burch Foundation has been a partner with us, and we're very proud to be working with them. And we also have announced a recent new program recently called WISE, Women in Entrepreneurial Science, that focuses on trying to really attract women in particular into biotech. As you probably know, there's a dearth of women leadership in biotech, and this has been widely commented upon. The question is, what do you do about it? And our feeling is, well, let's really encourage women who have an interest in biotech, but maybe don't quite know how to get into it, by giving them the resources and the guidance that they need early on so that they become tomorrow's entrepreneurs and future CEOs.
What types of problems do you think are best suited for a lab like yours or an institute like yours versus a startup versus more incumbent businesses? Or do you have a view on that, or do you try to approach that in a strategic way, or is it more organic?
Well, it's a hard question, I find. This is one of the hardest questions, and I'm always interested in how people try to answer it, because what I've found is that I used to think it would be obvious when you had an idea or a discovery that was ripe for a company to either license that technology or license that intellectual property, or to start a new company to build it out. But I found that it isn't always obvious. It's not always clear. Am I really at a point in this research where I've got enough here that I can really start to build a focused team, or hand this off to a team and they're going to be able to run with it? Or is it the case that there's still more innovation that's needed? There's still more kind of, casting about, turning over rocks, trying to figure things out. And, so you try to make your best guess with that. And hopefully most of the time you get it right. But, it really is a challenge, I've found. And I've heard this from other people too, that it's not always clear. Are we still innovating, or is this become kind of an engineering problem and we have all the pieces, we just need a team to put them all together?
Mm-hmm. When you're giving advice to people in terms of their careers, a lot of data scientists have early stage academic careers and then go on to work for companies. How do you give what guidance do you give to people in terms of thinking about staying in a research, a academic research lab versus going to a company or a startup, that kind of thing? Do you find that different personalities do better at different places? So.
I think they do. Yeah, I think they do. I mean, I guess first and foremost, I feel that my job as a mentor is to learn about my trainees and try to get to know them both as scientists and as people, so that I can help them figure out, what they want to do and where they're going to be most productive. And it's so interesting. It's happened to me again and again where I see people that come to the lab and they're clearly talented. Sometimes they don't get off on the right project right away, and there's some struggling that goes on. But as we get to know each other and I figure out kind of what they like to think about and the kinds of people they like to work with, I try to help them, get on the right track with what they're doing. And more often than not, when you get that right, not only does their science take off, but they also have more clarity about, what they're going to want to do in the future. And so often by the time they're ready to leave the laboratory, to leave my laboratory or our institute, they have a good sense. They've figured out that, well, I really, really like working at the bench. I don't want to write grants. I don't, write sometimes, or I don't want to be teaching classes. So, that they realize that they want to take a path that doesn't maybe involve sort of a traditional academic path, but other times the academic route is perfect for what they like to do. And so it's really just a question of, learning enough about the person so that you can help them decide, where they're going to be happiest and most productive.
A couple more questions from me, and then we'll open it up to the audience. I wanted to touch on the ethics of all of this. We work in artificial intelligence and machine learning where there's lots of ethical questions. You might actually work in the field where there's even more ethical questions. I'm curious how you approach that and think about it. Maybe we'll start there, and I'll have some follow-ups. But generally, how do you approach the ethical challenges of all that you're doing?
Well, it's interesting. I often think that machine learning and artificial intelligence is kind of the tech parallel to the CRISPR world in some ways, and that you're right, that I think there are some very interesting and big challenges in both of those arenas that we have to be aware of and as best as we can, grappling with as these areas advance, because they're both developing so quickly. In my case with CRISPR, so I'm a biochemist. I come out of a sort of fundamental science kind of background, and believe me, I never imagined that my work would eventually have sort of social, legal, bioethical implications, and have this sort of potential for societal impact like CRISPR does. And so I've had to really, grapple with that, not only with myself, but also asking myself, what's my responsibility as a scientist and as someone sort of involved in the origins of this field, do I need to get involved? And my answer, not without some struggle with myself, but my answer has been yes. I need to be involved in this, and I need to be, I need to be really publicly discussing the challenges that we're facing with CRISPR because of its extraordinary implications. And I think that, a couple of the areas of challenge that come to mind. One is human heritable genome editing, meaning making changes in human embryos that create changes in DNA that become part of the entire person and can be passed to future generations. So we're really talking with that kind of use of CRISPR about, engineering the human race, which is sort of an extraordinary thing to think about. And the other big area is environmental impacts, and using CRISPR in ways that could have profound impacts in the environment, certainly if misused. And so there's, I think in both of those arenas, again, there's a lot of expertise out there. And the question is, how do you pull those experts together, make sure they understand enough about CRISPR so that they can apply their knowledge to this field and this area? And I've been really fortunate that I've had a number of colleagues in academics, but also in companies who have been really willing to step up to the plate. We've had a number of the organizations around the world, whether they're scientific societies or the World Health Organization or UNESCO, that have gotten involved, and that's been a huge help. So I would say, at this point, we're at a place with CRISPR, at least, where there is kind of a global forum for looking at the pace of development in the field, and really asking the hard questions about should we someday be supporting human heritable genome editing? Is that a place that we should be going with CRISPR?
Does that forum or that group that's having those discussions, do you also try to influence lawmakers? And how do you think about the legal apparatus in both this country and in the world, and the influence it has on what's allowed and what's possible, and the interface there?
Well, it's interesting that at our institute, for example, we've been approached by a number of regulators from governments around the world, not just here in the United States, but elsewhere. And I've had several occasions to meet with those regulatory bodies, and to really get into they often ask fundamental questions about CRISPR, what it is, and how does it work, and where is it headed in the future, what's possible today, what's going to be possible in the future. And I would say I've been quite impressed with those groups. I think my sense is that they're sincere. They realize that they realize what they don't know, and they're really trying to educate themselves, and they recognize that this is a very rapidly advancing area of technology that needs attention. It needs their attention. The other comment I would make is that I think it's very difficult to have regulations that are applied globally. I think it's just for lots of reasons. It's really impractical to enforce such a thing, for example. And also, different cultures, different jurisdictions have different ways of approaching these kinds of challenges. And so I think that for me, I've really valued being able to talk with scientists in different countries, hear their views, try to understand their thinking. And I think we've been fortunate that with these forums, such as the one organized by the World Health Organization and another by the national academies, that this has really provided opportunities for these scientists globally to come together, and for companies to engage as well as they see fit.
So right now, do you think generally regulators are sort of hitting the right balance between innovation and caution?
It's always a struggle. I mean, I think right now I'm thinking a lot about the Food and Drug Administration, the FDA. How do we work with the FDA to ensure that there's a path forward that will, to the best of our abilities, provide for safety, but also efficacy of CRISPR, let's say, clinical therapies, but without undue delay. It's, and again, it's a challenge not just with CRISPR, it's with any new therapeutic development. I think with CRISPR, there's some unique challenges, in particular, the fact that CRISPR can be tailored to individuals. On the one hand, that's very exciting. On the other hand, how do we think about running clinical trials for something like that? It's sort of impossible. You can't really run a trial for each individual. So, we really need a partnership. And I've been quite pleased. We have a wonderful director of public outreach at the Innovative Genomics Institute, Melinda Kliegman, who has been working closely with the FDA and also with the USDA, the Department of Agriculture, because again, similar issues there when we think about applying CRISPR in agricultural products. And so, I think just having those ongoing discussions and debates, is very, very important. And what I've gotten from those agencies is that they appreciate that scientists in our team are taking the time, to talk to them, to tell them about what's happening in the field, keep them up to date with the latest in the technology, and really provide a forum for the kind of debate that needs to happen.
Well, as I mentioned in the beginning, this is an audience that's very interested in making breakthroughs, important breakthroughs. What advice would you have for all of us?
Well, I certainly think it's important to, I think we all have to pursue what we're passionate about doing, for one thing, because then that makes us really driven to do something if we're excited and passionate about it. And I think, innovation is, it's kind of an interesting combination of serendipity and opportunity, so I think for all of us that are trying to, continue to innovate on different things, there's always a, kind of looking for where are the opportunities? What are the big challenges right now? What are the big questions that need to be addressed that will really, break through if we can solve them or deal with them? And then you try to take those questions and look for where's the intersect with what I have expertise on, or the kind of team I have access to, the kind of resources I have access to. And when you get that right, again, in my experience, a lot of times, that's where the real breakthroughs happen, is that combination of opportunity and the serendipity that comes with taking a smart, team of people and focusing them around those opportunities, those fundamental problems that you're trying to break through.
Excellent. Let's take a few questions from the audience.
I'm very conveniently next to the mic, so I'll open the first one. Thank you, Doctor, so much. You talked a lot about individual cases of gene therapy. I'm curious your thoughts on using gene driving to release into the population, maybe in the one hand, let's say, forcing a harmful mutation into an invasive species that we introduce, so we're really getting nature back to its own, or in the case of mosquitoes in sub-Saharan Africa, where you're eliminating something that was already there but would save hundreds of thousands of lives. But once you release it, you can't really stop it. Your thoughts on that at a global scale?
Yeah, it's a great question. And you're referring to an application of CRISPR known as gene drive that basically engineers CRISPR in a way that allows it to transfer genetic information horizontally. In other words, without waiting for Mendelian genetics to take place, but sort of moving genes through a population quickly. And you gave the example of mosquitoes. That's a very interesting example, because there's been, as you probably know, there's ongoing work to use CRISPR to introduce either sterilizing mutations into mosquitoes, or mutations that prevent the spread of disease in various ways. And then on the one hand, extraordinary opportunity there in global public health potentially, but also a real risk in terms of making sure that we know what we're doing when we release something like that into the environment. So my feeling is that that's very important work that needs to go on, and it just needs to happen with real caution and an eye towards safety first. And I think right now, from what I've seen, groups that are doing this, that is how they're proceeding.
Hey, thank you so much for your time and for your work. You mentioned the situation where someone has like a flash of inspiration and then other people around them are telling them, like, what are you talking about? There's no way. How do you handle that in an organization? What do you do if someone comes to you like, I have this amazing thing, and you're like, that's a bad idea? What do you do in that situation?
Well, I guess if you're asking sort of more of a personal response to that, what do I do if somebody comes with an idea and I think my first thought is, oh, that's crazy, or that'll never work, or something. What I try to do is I try to step away from that reaction in myself and say, well, wait a minute, maybe they're onto something. What if they are? What if they're right? What if this is, and usually what I do is, if I think there's a kernel there potentially, I try to get them to flesh it out a bit more. Maybe they do some exploratory work to see if their idea gets any traction. So, because I'm really aware that, and I see this in myself, and I think it happens maybe to all of us at some level, that especially when you've been working in an area for a while, you can kind of have blinders on, right? You kind of, you find yourself thinking about things in the same way, but that could be limiting, and it could prevent you from seeing a connection or making, having an idea that really could be a breakthrough. So I try to fight it. And but if things really, if it seems like the idea really isn't impactful, those are the things that I try to really limit as much as I can. If I think an idea just isn't going after the key core questions that really we should be addressing, I try to point that out. Whereas if I think someone has an idea that's a little bit out there, but it's going after the right problem, then that's kind of in a different category for me.
Thank you so much for talking. I have a two-part question. You talked a little bit about the challenges with drug delivery, but what are some of the challenges that you see in our understanding of the genome that need to be surpassed before CRISPR can be more widely applied in genetic medicine? And then what are some of the data challenges that you face in genomic data, because the scale of genomic data can be so large?
Yeah, those are two really great questions. So for the first one, our understanding of the genome, I guess personally, I feel like the genome is incredibly complex. And just as a reminder of that, we still have, we have bacterial species where we have, whole genome sequences that are very small, because these bacteria are very, they have very compact genomes, where people have been able to pare it down to the essential genes that you have to have for this organism to survive. And a significant fraction of those have unknown function. And this is in a microbe, right? So it's just, and then you start thinking about the scale of, now how do I understand a magnolia or a human genome that's, many times larger and more complex. So it's humbling, it's always, I think it's important to keep that in mind. That being said, CRISPR is having a big impact there, because we can use it as a tool to probe the function of not only individual genes, but whole sets of genes. You can do this in, many different kinds of cells. You can use it to perturb individual genes precisely, and then ask what's the impact on a cell or a tissue when you do that, or even a whole organism. And so that's extraordinary. And then that kind of gets to the second question, which is what are the challenges with the data that are coming out, and what's going to be necessary to. This is another thing I think about a lot right now, is that I'm coming into the field of bioinformatics very much as an outsider, but it's clear that you have to have really high quality data, I think, to be able to get meaningful output. And so you can have the best algorithm in the world, but if you're querying a data set that is flawed in different ways, it's going to be very hard, I think, to get accurate insight. And so how do we do that? It's a big challenge, because right now, lots of academic labs are using CRISPR, but they're all applying it slightly differently and under different conditions, and the data they generate are, they're kind of one-offs. And so I've been in a number of conversations lately where we're talking about, could we pull together a large consortium that would agree that we're going to generate these kinds of gene knockout or gene perturbation data sets that will be generated under a standard set of conditions and produce a very high quality of data that can then be accessed. And then, of course, that really gets into your world of, where do we store the data? How do we make it accessible? How do we make it searchable easily? What format are we using? All of that. So it's a big challenge, but it's a good one, because I think there's a big window of opportunity there if we can figure that out.
We have time for, I think, two more, so here you go.
Yeah, thank you. I wanted to ask a little bit about the thought process behind developing an individualized solution for treating a specific individual's diseases versus a more generalized solution, and maybe some parallels that we can take out of your specific domain into what we do.
Right. Well, maybe I'll give an example. So, let's take the disease muscular dystrophy. This is a disease that, again, it's caused by mutations in a single gene. So in principle, it's a great kind of, entry-level target for CRISPR in that sense. However, if you look at the details of what mutations occur in patients that have muscular dystrophy, they're all over the map, right? They're really different. And so on one extreme, you could say, well, I'm going to design a CRISPR cure for each individual that addresses, the particular mutation that they have, versus I'm going to try to come up with a CRISPR-based tool that can correct a whole range of, we say, well, we're going to have one CRISPR tool that at least in 25% of the patients, it will work for them. And I think the latter is really the more practical approach. And then you have to figure out how to do that. And that gets into really into the details of what are the mutations, can we group them into different categories that could be, as a set addressable by different CRISPR tools. And I think that's a very interesting, future direction for CRISPR that will absolutely involve informatics. It will involve, the right kinds of algorithms that will allow us to, put those mutations into different buckets. And as we get better and better at predicting, which kinds of, iterations of CRISPR are going to be most effective with the least potential for side effects or toxicity, and that's happening more and more. I think we're going to see an intersection of that with the ability to also design these tools so that they will be able to address a whole set of mutations. And that's where the real impact is going to come in, because then you can start to imagine, back to this question of scaling, right? How you could really scale a technology like this to have much broader impact than if we try to go, individually one by one.
One more.
I think that some might argue that there are parallels in this question between CRISPR and machine intelligence. Especially given the clear lack of strength and enforcement in international law, do you ever worry that there's a sort of inevitability to an arms race of sorts, where some countries might take certain actions, in either of those fields, and others might feel like they have to override ethical concerns just to sort of keep up in the international stage?
Well, I guess I think, at least today, that situation really pertains more to machine learning and AI than it does to biology. And why is that? Well, I think it's still the case that our field is, there's enough unknowns about the way these technologies work in different biological systems, that it's still the case that there's a lot of work that has to go into any kind of application. And that's true whether we're talking about, editing yeast, or whether we're talking about editing the human brain or even human embryos. And so at some level, the technology itself is kind of it creates its own bottleneck, if you see what I'm saying. But at the same time, I fully, I appreciate the risks at hand, and I don't know how we deal with them except to deal with them. We just have to get out in front of these issues. I think that transparency is incredibly important. Having an international community of scientists. I think it's very interesting. We didn't talk about it earlier, but you may know that in 2018, a scientist in China announced that he had used CRISPR to edit the DNA of 2 human embryos that were implanted and to create a pregnancy. And those young girls were born with edited DNA. And I think the reaction to that announcement globally was very interesting. And it ultimately led to that scientist being jailed in China, and to I think a really strong negative international sentiment that, that is not where this technology should be going right now.
Dr. Doudna, that was very insightful, very helpful. Thank you so much for joining us. We really appreciate it. And thank you for all the work you do.
Thanks so much for listening.